I'm going to start because there is no time. نشوف كده بتاع شغال ولا مش شغال؟ شغال. Okay. I don't have time. We want to finish. We are going to finish now, okay? If you look again, aspartic acid, as I said, have a light blue color, will give the amino group. Will give the amino group to the previous amino acid, which is citrulline, to give me a complicated structure, which is called arginine and succinic acid. Arginine is an amino acid. Succinic is an organic acid. To bind these two huge structure, we need to break adenosine triphosphate into adenosine mono. If you look, I have first nitrogen, first amino, second amino, third amino. This carbon atom, which is attached to 3 amino group, is called guanido. Then a lyase enzyme. Lyase means breaking. I will break argininosuccinic acid by argininosuccinase to get arginine. Arginine is a semi-essential amino acid, which has guanido group. Carbon attached to NH, a second NH, a third NH. This is unique to arginine, okay? And we get fumaric. What are these? These are the citric acid cycle, okay? These are steps in the citric acid cycle. Arginine is broken by arginase. Arginase is found only in the liver. So all enzymes of urea cycle are found in other tissues except arginase. Arginase is found in the liver only. So the liver is the only organ in the body which sensitizes urea. This is urea. What is the structure of urea? Ketone, C double bond O, 1 amino group, a second amino group. From where the first amino group from ammonia, the second amino group from aspartic, okay? And then arginine, when it liberates urea, it will convert it into ornithine. Ornithine will return again to go inside the mitochondria. You have to study the cycle carefully with the name of enzymes. You will understand why I should memorize the name of enzymes. If you don't study the citric acid cycle, you are not going to study this step. So I'm not going to discuss it. I'm not going to explain. because these are steps in the citric acid cycle. Okay? Regulation of the urea cycle. The first enzyme of urea cycle is called CPS1. Bind carbon dioxide to ammonia. The first enzyme is called carbamoyl phosphate synthetase. Binding of ammonia to carbon dioxide and phosphoric acid. This enzyme needs adenosine triphosphate 2 molecule, which are broken into 2 molecules of adenosine dye. This enzyme needs activator, which is called N-acetyl glutamate. What is this compound? Glutamic acid with attached acetyl-CoA. N-acetyl glutamate is formed by an enzyme called N-acetyl glutamate synthetase. This enzyme needs arginine as an activator. So I need 2 activators. The first enzyme, which is called CAPS, the first enzyme, bind carbon dioxide to ammonia to sensitize carbamoyl phosphate. CPS1 is a mitochondrial enzyme, needs activator. What is the activator? N-acetyl glutamic acid. How can N-acetyl glutamic acid be sensitized? By a synthetase. This synthetase requires arginine for activation. Both glutamic and arginine comes from high protein diet. So you must take protein-rich diet in order for urea cycle enzymes to function. The first enzyme does not act except in the presence of glutamic and arginine. Okay? So this is one method of regulation. High protein diet increase the concentration of the enzymes of urea cycle by 10 to 20 fold. Okay? Administration of protein catabolic hormones. What are the protein catabolic hormones? If anyone takes cortisol as a treatment for any immune disease, glucocorticoid hormone or steroid hormone, it will make protein catabolism, and this increase the enzymes of urea cycle. Starvation. During the starvation, there will be breakdown and degradation of proteins. For any degradation of the protein, we should sensitize this urea. So the urea cycle enzymes increases during starvation. The most important thing in this lecture is hyperammonemia, because this is the diseases. The most important thing you have to study carefully is hyperammonemia, because hyperammonemia, ammonia is toxic to the brain and fatal. It leads to death. The plasma level of ammonia, this is not the plasma level of ammonia. The plasma level of ammonia is less than 0.05 milligram per deciliter. That's mean 50 microgram. 50 microgram. 0.05 milligram per deciliter. This is the blood ammonia. Don't mistake with the blood urea. Urea is non-toxic, so the level is high. 10 to 50 milligram per deciliter. طبعا 10 to 50 is much more than 0.05 milligram per deciliter. Urea is sensitized by the liver, go to the kidney to be excreted in the urine. In cases of renal failure, the kidney cannot excrete urea, so the blood urea increase. In cases of liver failure, liver disease, the liver cannot sensitize this urea, so ammonia level rises in the blood and there will be ammonia intoxication. Ammonia is toxic to the central nervous system. What is acquired hyperammonemia? Acquired means a disease in the liver which occurs in adulthood. A person who is born normally has no diseases in urea cycle, but he has got liver disease such as liver cirrhosis, hepatitis, inflammation of the liver, alcohol ingestion, your biliary stones, any biliary tract obstructions, any liver disease will open a collateral circulation. You are going to study collateral circulation in physiology, and the blood from the intestine usually go to the liver. This is called portal circulation. The blood which comes from the intestinal wall, absorption of food from the intestine will go to the portal blood. The blood will go to the liver, and then the liver will filter the digested foodstuffs which we absorb from the intestine, and then will secrete certain molecules to the systemic circulation. If the portal blood is translocated or shunted to the systemic circulation, there will be elevation of ammonia in the blood, because we have ammonia in the intestine, because the intestinal bacteria acts on proteins of the diet. So ammonia from the intestine will be absorbed and go to the portal blood, go to the liver. The liver does not allow ammonia to go to the systemic circulation. The liver converts ammonia into urea. So if there is a liver disease, ammonia will be elevated in the blood, and this leads to tremors like convulsion, slurring, interrupted speech, somnolence. Somnolence means desire for sleep, vomiting, cerebral edema, blurring of vision, coma, and death. All these are manifestations of hyperammonemia, and these are central nervous system toxicity. Again, tremors, like convulsions, involuntary movement, interrupted speech, slurring of the speech like any person who take alcohol, somnolence, desire for sleep, vomiting, cerebral edema means water in the brain, blurring of vision, unclear vision, coma, and death. So it is very serious to have hyperammonemia. What is a genetic or hereditary hyperammonemia? This is very important question in the exam. Hereditary means a newly born baby has mutation in any enzymes of urea cycle. So there will be hyperammonemia. Any enzyme, but very dangerous to have mutation in the first two enzymes, because the first two enzymes conjugate ammonia to carbon dioxide and conjugate ammonia with citrulline. So if we have defect in the first two enzymes, the level of ammonia will be toxic and the baby will die. If we have defect in the enzyme number 3, 4, 5, the level of ammonia will be mild elevation, not marked elevation, so the baby will not die, but he have troubles, central nervous system troubles. But all of these enzymes lead to hyperammonemia. The most common disease is ornithine transcarbamylase deficiency. This is the second enzyme. So the most common to occur in children, the most common to occur in children, or the incidence or the rate of occurrence is high in ornithine transcarbamylase. We may have defect in the first enzyme, carbamoyl phosphate sensitase. You have to know the name of the disease. Citrullinemia means accumulation of citrulline in the blood, elevated level of citrulline. The name of the enzyme, you have to study it. Argininosuccinate sensitase. Argininosuccinic acidemia means this acid is found in the blood due to deficiency of argininosuccinase. Elevated arginine level in the blood, hyperargininemia due to deficiency of arginase. This table has to be memorized. Of course, you will not be able to study or memorize unless you study the diagram. I suggest to study the diagram slowly and carefully and divide it along several days, along 2 or 3 days, to know what exact in the urea cycle, and then to memorize the name of the diseases and the defective enzyme. Okay? What are the treatment? The treatment is not written. Of course, there's no problem. The treatment are written in your book: to have low protein in the diet, to take benzoic acid, to take ornithine or arginine. All these substances help to excrete glutamine, to get rid of glutamine from our circulation. Okay? Thank you for your attendance. And I will stop there. Is everything clear, please? This person do not be admitted, and Compuland have to hear the video because he was admitted now and this is too late. Is everything clear? I don't say that citrulline is found in the liver only. I say that arginase is only found in the liver. Arginase enzyme is only found in the liver. Okay? Anyone want to ask any question? Please, if you don't understand anything, hear the video several times until you understand, and write your question on the WhatsApp group and I will answer it. Okay? Thank you very much for your attendance, and I'm going to leave now. Anyone want to ask me anything? Okay? Acquired hyperammonemia occur in liver disease. This is a question. Where does ammonia toxicity acquire? In liver diseases, in acquired liver diseases. Because in liver disease, the liver cannot sensitize urea, cannot remove ammonia from the blood, so there will be ammonia toxicity. CPS is an enzyme called carbamyl phosphate synthetase. CPS is an enzyme, the first enzyme of urea cycle called carbamyl phosphate synthetase. Any other question?
Doctor?
Yes.
There's a question that I've got before, that the net removal of amino acid to nitrogen is conducted. Where does it conduct it? Mainly where?
In all tissues of our body. Removal of amino acid nitrogen by transamination in all tissues. L-glutamate dehydrogenase is found in all tissues and organs in our body. Okay, all body metabolize amino acids, but synthesis of urea occurs only in the liver. Okay?
Okay. So
Okay,
the net
any
is
other question, please? Any other question?
Okay.
So I'm going to leave now. Thank you very much for your attendance. And any more question, please write it in the WhatsApp group. Goodbye. Goodbye.
Okay, thank you, sir.
Okay, thank you. Goodbye.
God bless you.
Okay.